a short history of cancer genes. Now, in the old days, cancers were recognized as new growths that expanded into normal tissues in a crab-like way. So they had little growths out that resembled crabs to somebody. In the 1600s, after the invention of the microscope, cells were first described, and the guy who called cells cells was Robert Hooke, you know, a contemporary of Isaac Newton's, and he called cells cells because they resembled the rooms that monks used to live in. And the cells he described were the cells in slices of cork, which is the bark of an oak tree, and those cells are actually dead cells, and cell death is what my research has been on. In 1775, Percival Pott identified the link between soot and squamous cell carcinoma in chimney sweeps. So these were the days when young boys would be sent up chimneys to clean the chimneys, and Percival Pott noticed that they often developed cancerous growths on their skin. And so that was some of the earliest evidence that the causes of some cancers can be explained by environmental factors. In the early 1800s, Raspail, Milne Edwards, Dutrochet, Purkinje, Valentin, Remak, followed by Schwann, Schleiden, and Virchow, developed cell theory. And that's the idea that all living things are composed of cells, and that cells are produced by cell division. And of course, we all accept that as an obvious fact now, that we were all once a single cell, and that divided in two, and so on and so forth. So cancers, of course, are made of cells, and they also originated with a single cell. In 1847, Virchow gave the name leukemia to leukemia, so it means white blood, and it's because the white blood cells are proliferating in an uncontrolled manner, and they begin to outnumber the red blood cells. So the blood more or less looks like pus. Treatment for cancers in the old days was, if any, was surgical, and the first radical mastectomy was in 1882. In 1886, the Brazilian ophthalmologist Hilario de Gouveia described an inherited retinoblastoma. So retinoblastoma is a cancer of the eyeball, the retina in the eyeball, and de Gouveia noticed it in 2 generations. So he had a patient who had a retinoblastoma, a tumor in his eyeball, and he removed the eyeball to this boy when he was young, but the boy had a family, and 2 of his daughters also developed, I think it was bilateral retinoblastoma. So it was clear to him that some cancers, or tendency to develop cancers, could be inherited. And then in 1902, after looking at chromosomes, so chromosomes are the DNA in cells where our genes are, and they're called chromosomes because they were first recognized because you could stain them with the stains that the microscopists would use. And this was many years before it was known that they were what carried out genetic material. Theodore Boveri proposed that tumors arise in a single cell that has damaged chromosomes. So he noticed that in tumors from some patients, the tumor cells had abnormal chromosomes, and all of the tumor cells had the same chromosomal abnormalities, whereas the normal cells in the patient didn't have those abnormalities. So, so that made him hypothesize that the tumors arose in a single cell, and that cell grew and gave rise to the all of the other cells in the tumor. Now, in 1911, Peyton Rouse discovered the first viral cause of malignancies, and that was in chickens. So there's a virus that was later called Rouse sarcoma virus that could induce tumors in chickens. And the way Rous did his experiments is he dissected out the sarcoma, which is a cancer of the muscle cells in a chicken, and he broke up the muscle tissue into small pieces. He ground it up with sand, and then he filtered it. And he filtered it so effectively that he knew there were no bacteria and there were no whole cells present. There was nothing that could be seen down the microscopes at the time. And so he passed it through a very fine pore filter. And then he injected that extract into young chickens. And those young chickens would also develop sarcomas. So he knew that what was causing the cancer was a filterable agent that was smaller than any bacterium and smaller than any cell. So that was the first evidence that some cancers, at least in chickens, could be passed on by viruses. So it's at least one of the causes of cancer. Now, in the following years, a number of other viral causes of cancer were identified. So in 1936, John Bitter, working at the Jackson Labs in Maine, discovered a virus that's now known as mouse mammary tumor virus. And it's known as that because some strains of mice would have an inherited predisposition to developing mammary tumors. And he, like Rous, took out those tumors and ground them up and made, initially he showed that the tumors could be spread both by inheritance along that strain of mice, but it could also be spread to other strains of mice that didn't spontaneously develop tumors if he fed those baby mice on milk from the cancer-prone strains. So he initially described a milk factor that allowed transmission of the mouse breast cancer by adoptive nursing. So once again, this was evidence that some tendencies to develop cancer could be passed down by inheritance, whereas others could be spread by infection. So this was a case of cancers being spread to young mice from a virus in their mother's milk. So we now know that both Rous sarcoma virus and mouse mammary tumor viruses are retroviruses. That means that the genome of the viruses is messenger RNA, just like the genome of coronavirus is an RNA. But these retroviruses have a peculiar lifespan in that they can exist in two forms. They can exist as a provirus, as a DNA copy in the genome of the organism, and if it's in the germline, it can be passed down through inheritance. But these, so that's the proviral DNA copy in the chromosomes of the organism, but they can also be transcribed into messenger RNA and packaged up into viral particles, and these viral particles can be infectious, and they can be spread through blood or other bodily secretions and infect other animals. But of course, the other animals that are infected, they'll only pass on the virus through inheritance if the provirus is inserted into the genome, into the germ cells. And another example of a retrovirus is the HIV virus. It can also exist in two sites, integrated into the cell's DNA, and also as an infectious viral particle. In 1966, 55 years after he described the Rous sarcoma virus, Peyton Rous was awarded a Nobel Prize. And there haven't been very many Nobels awarded for cancer research, but there have been a handful. One was in 1926, and that was to, I love the name, Johann Andreas Grib Fibiger, for his discovery that stomach cancer was caused by the roundworm Spiroptera carcinoma. This is, of course, not true. and it was later described as one of the biggest blunders made by the Karolinska Institute. Some people argue that another big blunder was the Nobel Prize that was awarded for the guy who invented the frontal lobotomy. In 1966, Charles Bretton Huggins got a Nobel for his discoveries concerning hormonal treatment of prostatic cancer. And in 1989, a Nobel was awarded to Harold Varmus and Michael Bishop, and we'll hear more about them later in the talk, for their discovery of the cellular origin of retroviral oncogenes. So once again, they're hearkening back to retroviruses that like the Rous sarcoma virus and the mouse mammary tumor virus. In 2008, Harold Zuerhausen was awarded a Nobel for his discovery that cervical cancer is caused by spread of the human papillomavirus. And of course, we give great credit to Ian Fraser for being one of the people to come up with a vaccine against human papillomavirus that hopefully one day will eliminate cervical cancer around the world. So once again, this illustrates that there was a lot of focus in the early days of cancers being caused by viruses, but most of the examples of that were from animals rather than humans. In 1958, Howard Temin and Harry Rubin showed that Rous sarcoma virus could transform primary chicken embryo fibroblast cells, so in tissue culture. So it was in the 50s that techniques were developed to grow cells in tissue cultures in petri dishes, and because they could lay out a lawn of cells, they could add viral extracts, and then they count the number of transformed colonies that would develop, and so they could work out the number of viral particles that were needed to transform the cell. So this shows some cells that are normally sitting down flat on the plate, and they don't overlap each other. They have this sort of cobblestone-like appearance you see down below. But if you added the Rous sarcoma virus to the cells, this retrovirus to the cells, then you get these foci of cells where the cells are all growing on top of each other in a higgledy-piggledy fashion. And by electron microscopy, you can see that they've got this spindly-like appearance. Furthermore, if you took these cells, or when they took these cells and injected them into chickens, these cells would form tumors, whereas the normal fibroblasts wouldn't form tumors in recipient chickens. In 1970, Steve Martin made a temperature-sensitive form of Rous sarcoma virus, and this one was able to transform cells if you cultured the cells at 37 degrees, but if you elevated the temperature of the culture, then the cells reverted their phenotype and looked like normal cells again. So these experiments, and so the hypothesis was that there was a protein that was being made by the virus that transformed the cells, but when you elevated the temperature up to 41 degrees, that protein became misfolded and no longer had its transforming activity. So these experiments showed that the remainder of the virus could still propagate, but that part of the virus that transformed the cells was likely to be a single gene product, and they called it the SRC or SARC gene. And this SARC gene wasn't required for viral replication, but it was the single gene that caused transformation of normal cells into malignant-looking cells. In 1971, after lobbying by Mary Lasker, and there's Mary Lasker with this lovely hairdo there and the pearls, she was a major lobbyist who convinced Richard Nixon to declare war on cancer. And that greatly increased the amount of funding to find out what really is the cause and what are potential new cures for cancer. Because up until then, the only thing that could really be done was surgery on some sorts of cancers. There was development of early forms of chemotherapy leading on from discoveries that radiation could kill some tumor cells in animals, and it also some of the agents that developed sometimes as nerve warfare agent or alkylating agents from mustard gas, they could also be used and were developed into treatments for cancer. But it was the boost of money from Richard Nixon's war on cancer that really led to the expansion of the cancer research effort. You know, up until then, it was generally believed that cancers in humans were due to either emotional stress, neuroses, or by viruses, environmental carcinogens, or radiation. But the basic biological causes and the mechanisms by which these agents cause cancers weren't known. In 1976, Dominic Stalin, who was working with Mike Bishop and Harold Varmus, used DNA hybridization, and that's where you take two different strands of nucleic acid and you add them together, and you can measure their hybridization temperature, and you can incubate them together at different temperatures and run them on a gel and work out how similar they are by their ability to hybridize to each other. He used that technique to demonstrate that this viral Sark gene from the Rous sarcoma virus was derived from a cellular gene. So there was a cellular gene in the normal genome of birds and also mammals that had very similar sequences to the viral cancer gene. And so this was a big shock, because why would it be that normal healthy animals would have these potential cancer genes within them? And if they did have these cancer genes, why weren't they developing cancer? What was the difference between the viral copy of the cancer gene and the normal cellular copy? So this was a really puzzling but major breakthrough. So in 1989, the Nobel Prize was awarded to Bishop and Varmus, and this, of course, caused quite some consternation to Dominic Stalin, but it was awarded to Bishop and Varmus for their discovery of the cellular origin of the retroviral oncogenes. So the hypothesis was that somehow these viruses are able to capture a normal, potential oncogene and activate it or switch it on, and that way they could become cancer-forming viruses. So retroviruses were subsequently shown to cause cancer by at least two different ways. One way is by inserting into the DNA, the normal genomic DNA, and if the virus inserted behind a growth gene, for example, then the viral regulatory sequences could turn on the growth gene and stimulate the cell to keep growing. And viruses, these retroviruses, can also pick up a cellular gene and then carry that gene like VSARC and use it to infect other cells. So many proto-oncogenes, so these are normal cellular genes that have normal functions in the body, were discovered by looking for homologs or the equivalents of retroviral genes that were known to cause retroviruses that are known to cause cancer in different animals. So these genes, ERB-A, ERB-B, ETS, FOS, JUN, MIB, CSFR, like, for example, CSFR is the colony-stimulating factor receptor. Colony-stimulating factors are white blood cell growth factors. And so if a virus can pick up that gene and carry it into other cells, it could allow white blood cells to make their own growth factors or respond to autonomously and thereby help them turn into a leukemia. And one of these genes is the c-MYC gene, and we'll talk a bit about that as we go along. Because many of the cellular homologs of these viral genes were involved in cell proliferation, for example, as growth factor receptors or signal transduction kinases that send signals from the cell membrane into the nucleus, and transcription factors that regulate the expression of genes, the idea arose that cancers could be caused by increased or unregulated activity of growth genes or the proteins they encoded. So one way that you can turn on one of these growth genes that normally exists so that you can grow and heal and repair your own tissues, one way that they can be turned on is if a virus lands behind them. Another way they can be turned on is if they're mutated by some sort of mutagen, such as radiation, for example. So it was only in 1956 that Tijou and Levin showed that humans have 46 chromosomes. And I was really surprised to hear that. Now, I imagine everybody here knows that we have 46 chromosomes, and we got 23 from our mothers, and then we got 23 from our fathers. But it was only as recently as 1956 that that we knew how many chromosomes humans had. Now, prior to that, it used to be thought that humans, I can't remember whether they had 48 or 44, but the number was only settled at 46 when Tijou and Levin used more sophisticated chromosome banding techniques and dyes, and actually nailed down what the number was. Because up until then, you know, somebody had said that the number was this, and then everybody else just confirmed it and didn't really bother checking. So each chromosome in our cells, and every one of our cells, except our red blood cells that have lost our chromosome, every one of our cells has 46 chromosomes within them. Again, 23 inherited from our fathers, 23 inherited from our mothers. Each chromosome is a single double-stranded helix of DNA. And laid out, if you stretched out each one of those chromosomes and stuck them all together, the 46 chromosomes in each cell would be a total of 2 meters long. Right, so they've got to be obviously be wrapped up and so on. But it's amazing that a cell can do that and can make copies of the DNA, and that you know, and now sometimes they break and sometimes they're joined around the wrong way, as we'll get into later on. But the human genome has about 6 billion DNA letters, and these are the GATC that this building is named after. And during cell division, every one of these 6 billion DNA letters has to be copied. And because in our bodies, over 1 million cells complete cell division every second, you know, and that's happened more or less since the day we were born, each of us makes over 2,000 kilometers of DNA every second. And that, of course, is useful to the forensic, police and scientists, because, you know, there's DNA coming out all over the place, and maybe it's not so surprising that if you touch a cup or a cigarette, that somebody can take copies of your DNA even from that small sample. So it's amazing to think, you know, in fact, if you stretched out all of the DNA in each one of our cells, all of those roughly 10 to the 13 cells in our bodies, each one has 2 meters of DNA. That DNA would stretch to about 1% of a light year. So it's an amazing amount of DNA, and every second, 6 by 10 to 15 letters have to be copied in our bodies. And if there's a mistake in that copying, it can potentially cause a mutation that, if it occurs in the wrong place, can cause a mutation that leads to development of cancer. Now, in 1960, so this is only, you know, a couple of years after they knew how many chromosomes humans had, but in 1960, Peter Knoll and David Hungerford reported that when they looked at the cells of patients with chronic myeloid leukemia, or CML, they found that there was this minute chromosome. And when they looked at the cells, other cells from those same patients, they didn't see this, this minute chromosome. And they called the chromosome the Philadelphia chromosome because they were working in Philadelphia. A few years later, Janet Rowley showed that the Philadelphia chromosome was made by a translocation at the tip of chromosome 9 onto the end of chromosome 22. So I'll just explain what a chromosome translocation is. So here are the 46 chromosomes laid out, and you can see there's 2 chromosome 1s, 2 chromosome 2s, 2 chromosome chromosomes 3, etc. And you can see this must be from a cell of a male patient, because there's an X chromosome and a Y chromosome. And these chromosomes have, the way you see them is you add a colchicine or a poison to the cell. So this would have been a leukemia cell. You add a poison that halts the cells in metaphase when they're dividing and the chromosomes are all condensed like this. And then you put the cells onto a hot plate and the cell kind of explodes, and then you stain the cells and each chromosome, and then you take photographs of it, and you, with a pair of scissors, you cut out each chromosome and lay them out. And by comparing the patterns of banding, you can tell the chromosome 1 from the chromosome 2, etc. So you can see these two chromosome 1s have a sort of a light grey band here, and then they have a dark grey and then an even darker grey, so you can easily match them up. And it was this process that allowed them to detect that, you know, humans have 46 chromosomes. And when Janet Rowley looked at the cells that had this Philadelphia chromosome, here's the Philadelphia chromosome down here, and she realized that an end of one of these chromosome 22s was now stuck on the end of chromosome 9. So that was the explanation for where this minute Philadelphia chromosome came from. It's kind of a deleted, shortened copy of chromosome 22. But the real interest comes from so what's happened is that in the cell, there's been an accidental break on the end of chromosome 22 and the end of chromosome 9. And again, these are single molecules of DNA. The cell has tried to repair them together again, but it's accidentally repaired the two broken chromosomes the wrong way round. And so now there's a little bit of chromosome 9 on the end of chromosome 22 and a little bit of chromosome 22 on the end of chromosome 9. So here's this shortened Philadelphia chromosome down here, and it's got a little bit of chromosome 9 in blue at the end. So this is called a chromosome translocation. And what can happen is at the break point, you can get a fusion gene occurring, you can get a fusion protein occurring, or you can have the regulatory regions from one gene, instead of turning on the gene that is meant to be next to it, can turn on a gene that was on a completely different chromosome. So the close correlation between chromosome, the Philadelphia chromosomes and chronic myeloid leukemia suggested this isn't just luck, it must be meaningful, and maybe there's some sort of oncogenic event that occurs because of this translocation. Now, in 1980, Owen Whitty showed that the retroviral oncogene, so now we're back talking about retroviruses, some retroviruses carry a gene called V-ABL, and Wendy Cook, who used to work at the Walter and Eliza Hall Institute, she did a lot of work on the Abelson virus that carries the V-ABL gene. But Owen Whitty showed that this V-ABL gene encoded a tyrosine kinase, and this is a kind of enzyme that's been implicated in a lot of signal transduction growth regulatory pathways within cells. And then 2 years later, Nora Heisterkamp showed that in chronic myeloid leukemia, there's involvement of a cellular equivalent of the viral ABL oncogene that was on chromosome 9, but it's been now stuck on the end of chromosome 22 right at the break point. And it's now known that this chromosome translocation leads to activation of the cellular Abelson gene, activation of that tyrosine kinase, so that the tyrosine kinase ends up being sent to the wrong compartment in a cell, and it's constitutively switched on to cause growth of the cells. Now, as I mentioned, the viral ABL and the cellular ABL gene encodes an enzyme. So if you want to and the cancer is caused by too much activity of this Abelson enzyme. So if you wanted to treat the cancer, what you'd want is an inhibitor of that enzyme. And indeed, in the late 1990s, Brian Drucker showed that an inhibitor of that ABL tyrosine kinase that was called Gleevec originally, and is now called imatinib, would cause CML cells to die. Now, chronic myeloid leukemia used to be a death sentence. Occasionally, some patients could be saved by having a bone marrow transplant. But nowadays, patients take this tyrosine kinase inhibitor, imatinib, and they more or less have a normal lifespan, although they have to keep taking the this drug more or less for the rest of their lives. So they're probably not cured of the cancer, but it's sufficient to stop any of the clones of cells bearing that tyrosine kinase to escape and cause disease. Now, in the mid-1960s, and we're going back to these retroviruses, Jenny Harvey isolated a transforming retrovirus from leukemic rats, and she found that these rats were capable of causing muscle tumors or sarcomas when injected into mice. And so this virus was named Harvey murine sarcoma virus. And so this is 1964, and it's to introduce the gene carried by that virus that was later called HA-RAS for rat sarcoma gene. There we go. Harvey murine sarcoma virus was later shown to be the result of a combination between a proviral genome, so the original virus caused, the parental virus caused tumors only very slowly, but after the virus has picked up this sarcoma gene, the RAS gene, then this retrovirus could cause tumors much more rapidly, for example, in mice. And so the cellular normal rat gene was called Harvey-RAS or HA-RAS, with the name coming from Harvey from Jennifer Harvey and RAS from rat sarcoma. And it was responsible for the transforming ability of the virus. Now, much later it was shown that the normal HRAS protein is another kind of enzyme that acts again in growth factor signaling pathways, and it can go from its and the RAS protein can be in an on state or an off state, but the mutant form of the protein that existed in the retrovirus is always in the on conformation, and so gave a constitutive growth signal to the cells that were infected by or carrying the virus. Now, another technological breakthrough was in 1973 when Graham and Van der Ebb developed techniques to transfer DNA into cultured cells. Now, I mentioned before how it was the assay that Temin developed that allowed them to put viruses and infect cells in tissue culture. Well, a similar technique was developed by Graham and Van der Ebb to transfer naked DNA into cells, into cultured cells in Petri dishes. Bob Weinberg used this technique to screen for genes with cancer transforming potential. So what he did was he took DNA from cells that had been chemically transformed, and that means these were cells that had been exposed to a cancerous mutagen, and so they'd selected cells that had transformed phenotype, so they were growing on top of each other, and then he would isolate the DNA from those chemically transformed cells and transfer it into to cell lines that were not malignant, and some of those cells would then form colonies and clusters of transformed malignant cells. So this indicated that the transformed growth properties of cancer cells is due to genes. It's due to the DNA that had been taken out of the original set of cells and had been transferred into the cultured cells. In 1982, Bob Weinberg's lab, but also Cooper and Barbacid labs, cloned a gene from bladder cancer cell lines. So they took DNA from human bladder cancers and they tried transfecting that DNA into cultured cells, transferring that DNA to cultured cells, and again looked for colonies that had malignant phenotype. And then they took those colonies of mouse cells and then extracted the DNA again, and then they went through multiple rounds, and then they looked for markers that showed that the DNA that was carried over, as it became more and more diluted for just passenger genes, they looked for the human genes that had originated presumably from the human bladder cancer. And when they isolated that gene that was responsible for transforming the cells, it turned out to be a mutant form of the human RAS gene. So the same gene that was originally known because it was found in these murine viruses. So once again, things, it was a very big sort of ha-ha moment, because the same gene that was associated with bladder cancer in humans had been identified as a cancer gene in rodents. So it showed that mutant forms of RAS could oncogenically transform mammalian cells. Now, mutant RAS genes are frequently found in a lot of other sorts of tumors. Pancreatic cancers, almost all pancreatic cancers have a single point mutation in a RAS gene. Bladder cancers, colorectal cancers, and many other sorts of malignancies. Now, another bird retrovirus was called MC29, and it was also isolated in 1964 in Bulgaria, and it was known to cause leukemia in chickens. And the sort of leukemia was called myelocytomatosis, and that's why they gave the gene that was eventually identified within it the name MYC, for MYC, for myelocytomatosis gene. So this virus could cause cancers when injected into chickens. In 1977, Duesberg and Vaux showed that this virus had a unique transforming gene that was not present in other related avian viruses, and they called it VMYC, again naming it after myelocytomatosis. So once again, the retrovirus can exist in sort of benign, slowly transforming form, and these are ones that don't carry an oncogene but have the potential to, when they insert, to activate an oncogene, or they can come in a potent form where the retrovirus itself has picked up the cancer gene and it can carry it into other infected cells. And once again, retroviruses can exist as proviral copies, and if they're in the germline, they can be passed on generation after generation, or they can be produced as infectious viral copies. In 1981, Hayward, Neal, and Astron showed that the endogenous version of the MYC gene, which is called c-MYC, so C stands for cellular, the cellular copy of MYC, whereas the viruses have the V designation, so the viral MYC. They showed that the cellular endogenous version of MYC could be activated by retroviral insertion. So if you don't have to mutate the MYC gene. If a virus lodges behind the MYC gene, the viral promoters and enhancers can turn on the cellular gene, and that can lead to a transforming activity. So these experiments showed that retroviruses could pick up an oncogene, or they could activate an endogenous copy of an oncogene. Now, in August 1982, Grace Shenong in Michael Cole's lab showed that the c-MYC gene, so the normal MYC gene, was translocated onto the Ig locus in plasmacytomas in mice. So to explain that, we're now talking about another example of a translocation, but this isn't the translocation that generates the Philadelphia chromosome, involving chromosomes 22 and 9. This is two different chromosomes. One of the chromosomes, I think it's chromosome 14, carries the immunoglobulin locus that normally produces the antibody genes. And in most plasmacytomas, and these are cancers of the antibody-producing cells, in most plasmacytomas in mice, there's a translocation that involves the Ig locus, but Grace Shenong showed that the translocation involved, the other partner of the translocation involved the c-MYC gene. And so the hypothesis was that there was one original B cell whose job was to make lots of immunoglobulin from the Ig locus, but because of this break and then an incorrect repair, that cell was producing, instead of lots of immunoglobulin, it was producing a lot of c-MYC, and the product of the MYC gene binds to DNA and drives proliferation. In December 1982, Carlo Croce's lab and Phil Leader's lab reported that the c-Myc gene was translocated onto the Ig locus. So this is an analogous translocation, but instead of in mice, this was in a cancer of the B cells in humans called Burkitt's lymphoma. So this showed that the biology was very similar between these oncogenic events in mice causing B cell leukemias or lymphomas, and in humans. So once again, this is a very striking, because it's another example of a translocation causing a leukemia in humans, just like in Philadelphia chromosome and chronic myeloid leukemia. In 1984, Tim Stewart and Phil Leader made the first oncogene transgenic mice, and these were called oncomice, and they were patented, and that created a lot of unhappiness in the cancer field. But anyway, they made the first oncogene transgenic mice, and they used as the oncogene c-Myc, but they put it under control of retroviral promoters, and these were promoters from the mouse mammary tumor virus, and these mice developed tumors. So here's a mouse here, and you can see it's got a breast tumor, and that's because the mouse mammary tumor virus turns on genes within the breast tissue of the mice. And these experiments show that a constitutively deregulated and switched on Myc transgene, and the transgene is just an extra gene that's been transferred into the genome of the animal, could act as a heritable predisposing factor favoring the accelerated development of clonal tissue-specific adenocarcinoma. So these mice all inherit the extra gene, the extra transgene, but they don't all develop breast cancers from birth. They for them to grow, they need activation of the gene by the hormones that activate the MMTV promoters. But it also indicates that just having the extra gene alone is not sufficient to cause tumors, because the tumors that developed were also clonal. So it suggests there's more than one oncogenic event required to turn a normal cell into a cancer cell. Now, in 1985, in a collaboration between Walter Eliza Hall Institutes and University of Washington in Seattle, another strain of Myc transgenic mice were made, and these ones were to try to make a model of human Burkitt lymphoma, or a model of these mouse plasmacytomas. And for these mice, rather than having the Myc gene driven by mouse mammary tumor virus promoters, the Myc gene was driven by the immunoglobulin regulatory regions. So the Myc gene would only be switched on, but in a constitutive way, in the B lineage, the antibody-producing lineage of the cells. And indeed, these mice are known as emu-Myc mice, and all of these mice, transgenic mice, develop lymphomas. Now, these mice showed that the incidence of cancer isn't simply a matter of nature, meaning the genes, and nurture, the environment. So emu-Myc transgenic mouse littermates, so these are, if they're the same gender, they're genetically absolutely identical. They're all on an inbred genetic background. They all have, the same transgene, and they have the same environment, because these mice are housed together. They have the same parents. They're in the same cage. They have the same food, and so on. All of the emu-Myc transgenic mice will develop lymphoma, but they don't have lymphomas at birth. And by about 10 weeks of age, roughly half of these emu-Myc transgenic mice develop lymphoma, but it's impossible to tell from the start which 50% of those mice will develop the lymphoma and which ones won't. And furthermore, you can tell that each of the tumors is a clone of cells, even though the mice would have billions and billions of B cells, all expressing the transgene. Only one of them has turned into a lymphoma. Now, after another 10 weeks of age, half of the remaining mice develop lymphoma, and after another 10 weeks of age, half the surviving remaining mice will develop lymphoma. So this suggests there's something stochastic and random occurring. So just having the the cancer gene, an active cancer gene, isn't enough to cause a tumor, there's something random that has to occur as well. So therefore, it was concluded that constitutive expression of c-Myc alone is not sufficient to cause cancer, to turn a normal cell into a malignant cell, but another apparently random event is required. And so presumably it's a random event is some sort of random mutation that happens maybe when the cell is copying its DNA. Remember, we've got to copy, 6 billion, or the mouse has to copy 6 billion DNA letters every second as every cell divides. And if a mistake is made, then it could cause a mutation that would complement the MYC gene. Now, we're going to change tack now and talk about tumor suppressor genes. And so I'll introduce Henry Harris. Henry Harris was born in Russia in 1924, but came to Australia at age 4. And I don't know how many people in the audience have ever heard of Henry Harris. You heard of Henry Harris? You would, Ashley? He's famous, yeah, good. But he ought to be more famous because he was the guy who provided the first experimental evidence for tumor suppressor genes. He graduated in medicine, I think it was up in Sydney, and then he went to Oxford and did a doctorate with Howard Florey. He discovered that another sort of virus, Sendai virus, could be used to fuse cells together. So you could take a cell of one type and a cell of another type and add the virus and make a cell that fused the cytoplasm and had 2 separate nuclei. In 1969, he carried out experiments where he took malignant cells and he fused them using the virus to normal cells, and he tried to find out, would the malignant phenotype be dominant over the normal phenotype, or would the normal cell be able to suppress the tumor phenotype? And he showed that there were some chromosomes that could, in the normal cells, that could suppress the growth of the cancer cells. And then if he kept culturing the cells, some of them threw out particular chromosomes and reverted their phenotype and became transformed once again. And it was always seemed to be the same particular chromosome that was lost. So he correlated the return of malignancy with the loss of a specific chromosome of the normal partner cell. And this provided the first clear evidence of the existence of a class of tumor suppressor genes. Now, after studying familial cases of retinoblastoma, remember I mentioned retinoblastoma at the start of the talk, it's this cancer of the eyeballs, and it was known back in the turn of the century that it could be inherited. But in 1971, Alfred Knudsen hypothesized that some cancers require 2 genetic hits that mutate both copies of tumor suppressor genes. So he thought that if there's a tumor suppressor gene, so let's call it RB because the disease is retinoblastoma, if you've got just one normal copy of the gene, it's a tumor suppressor gene, so it'll stop tumor growth. So if you just have one hit and you mutate one copy or delete one copy of the gene, you've still got a spare from your other chromosome from the other parent, so you won't have any retinoblastoma. But if you have a second hit and it's unlucky enough to be on the other copy of the gene, then you'll develop retinoblastoma. So, the chances of having getting a spontaneous mutation to the retinoblastoma gene, let's say it occurs in 1 in 100,000 people, the chances of it occurring to the same gene again will be that squared. So it might be only 1 in 10 million people that spontaneously develop retinoblastoma. But if you inherit one dud copy of the retinoblastoma gene, then you've only got one spare to go. And so, if you've got that, then there's a chance that you'll lose the other copy. And so people who've got one dud copy of the gene have an inherited tendency, increased incidence to develop retinoblastoma. So once again, this is called the 2-hit hypothesis, that for sporadic cancer, you need 2 hits, which is very unlikely. So that's why sporadic retinoblastoma is very rare. But if you have the inherited tendency, you've already got one dud copy, so it's much more likely you'll develop the disease because you only need one extra hit. So in 1986, 2 groups cloned the gene for retinoblastoma, and it was one of the first tumor suppressor genes that was cloned. Now, in the 1960s, it was shown that monkey polyomavirus SV40 was oncogenic in rodents. So it's putting a monkey gene into mice and they develop tumors. And it was shown that the virally encoded protein called the SV40 large T protogene was required for this transformation of the mouse cells to cause tumor in the mice. Now, David Lane looked for proteins within the cells that could bind to this viral protein, and he found that there was a protein that had a molecular mass of 53 kilodaltons, and so it was called P53, and it was found that it could bind to this SV40 large T protein. So it was hypothesized that it might play some role in causing the transformation of the cells. In 1982, the P53 gene was cloned by Georgiev, and initially it was thought that P53 was an oncogene and that somehow SV40 was activating P53 and that was causing tumors. But it turned out, and this was shown in 1989, that P53 was actually a tumor suppressor gene rather than an oncogene, and it suppressed tumors, but the mutant forms of P53 could act as in a dominant negative fashion and to inhibit the normal copies of P53. So mutated P53 could act as a tumor activator by suppressing the suppressing activities of P53. And once again, we'll go into experiments in nature where there was a syndrome called Lee-Fraumeni syndrome described by Frederick Lee and Joseph Fraumeni, who were studying pediatric and familial cancers. So they were studying some inherited tendencies to developing cancers. And these were cancers not of the eyeball in this case, but of all different sorts of tissues, leukemias and soft tissue tumors as well. And so that was called the Lee-Fraumeni syndrome. And the thought was, well, presumably there's some other tumor suppressor gene that's involved, and the people with an inherited tendency already had one mutant copy of the gene. And in 1990, it was shown that it was that P53 gene that was the cause of Lee-Fraumeni syndrome, and that was work from Stephen Friend. Now we're going back to looking at another translocation, and again looking at Janet Rowley. Janet Rowley, remember, was the one who showed that it was the chromosome translocation between chromosome 22 and chromosome 19 that caused chromosome 9 that caused the Philadelphia chromosome. So she thought, well, look, if I look at other leukemias, maybe I'll find other translocations, and that'll allow identification of novel cancer genes. So here's chromosomes from a patient, and this one's obviously a woman because you see the two X chromosomes. And in here, Rowley noticed that there was abnormalities in chromosome 18 and chromosome 14. So a little bit of chromosome 18 stuck on the end of chromosome 14 and vice versa. Now, this is in 1984, the T1814 breakpoint was cloned by Yusuji Tsujimoto, and the gene at the site was called BCL2, and that lab also looked at other translocations and called them BCL3, BCL4, BCL, and so on, up to at least BCL6. And the gene at the site, so here's the translocation here. It's a little bit of chromosome 14 stuck on the end of chromosome 18 and vice versa. And this translocation, like with Burkitt's lymphoma that I mentioned before, sticks something on the end of the immunoglobulin locus. So the cells that have this accidental misrepair think that they're producing lots of antibodies, lots of immunoglobulin, but they're producing lots of this protein called BCL2. So the initial thought was, well, maybe BCL2 is going to be a growth gene like MYC, and it's going to cause the cells that have the translocation to proliferate and proliferate and proliferate. But in 1988, it was shown that BCL2 did not function like other cancer genes because it did not stimulate the cells to grow and proliferate, but it only acted to inhibit the cells from being able to kill themselves. I'll explain a little bit about that later. So it was known, or was previously known, that growth factors landing on their receptors could stimulate cells to proliferate, and they'd also survive. And if you took away growth factor, then the cells wouldn't proliferate, and they would also die. And they would die by activating an endogenous cell suicide mechanism. But these experiments where BCL2 was put into cells suggested there are independent pathways within the cell that regulated cell death and proliferation. So if you put lots of BCL2 into a cell, like if you have a normal cell and you take away growth factor, there's a default activation of this cell death pathway leading to programmed cell death or apoptosis. If you turn on BCL2 in the cell, you don't get any proliferation if you don't add growth factors, but BCL2 can somehow stop the cell from activating its default death pathway. If you add growth factor to the cell, it's able to proliferate and it still survives either because of growth factor or because of BCL2. But the conclusion was that there are separate pathways downstream of the receptor that independently regulate this default cell death pathway to inhibit it, but also to stimulate proliferation. So there are distinct pathways controlling cell division and cell survival. BCL2 specifically inhibited the cell suicide pathway, but didn't stimulate proliferation. Inability of cells' ability to kill themselves can lead to cancer. So this was a deduction, because remember, BCL2 was discovered because it's activated in cancers in humans, namely follicular lymphoma, and also known to be activated in chronic lymphocytic leukemia. But it's also known that blocking cell death alone couldn't transform a normal cell into a malignant one. So I've been mentioning about programmed cell death and apoptosis, and so there's another nice Australian link to that, because the term apoptosis was coined by John Kerr from the University of Queensland, where he was a professor of pathology. Now, he was working on this physiological process of cell death, but he went on sabbatical in Aberdeen, University of Aberdeen, where he realized that the cell death that he was observing was similar to the cell death that was being studied in Alistair Curry's lab. So they decided to give this physiological cell death phenomenon a new name, and they called it apoptosis. So they decided that the way the body makes a new cell is by mitosis, or cell division, and the way the body gets rid of an unwanted cell is by this process they called apoptosis. So Kerr wrote that both apoptotic bodies, so cells killing themselves, and mitotic figures, or cells dividing in two, are sometimes numerous in rapidly growing tumors. But it's the balance between the two processes that determines the rate of enlargement. So if you have more cell death than cell growth, cell division, the tumor will wither away. But if you have more cell division than cell death, then the tumor will get larger and larger. But he also noticed that it occurs spontaneously in untreated malignant neoplasms and participates in at least some forms of therapeutically induced tumor regression. So they knew from experiments that Curry was doing that when you added steroids or some hormones to cells, that would activate cell death. They also knew that if they castrated male rodents, that they would see a lot of apoptosis in the testes of the mice and the prostates of the mice. So this shows the number of publications per year around the world that mention the terms apoptosis or programmed cell death. And you can see the term was adopted by Kerr, Wiley, and Curry in 1972. The term programmed cell death was first used in 1965 by Lockshin, looking at cell death in insects. But basically, nobody was really interested in it. But in the late 1980s, suddenly it took off. And the reason it took off was because the mechanisms for programmed cell death or apoptosis were first identified, and then the first gene involved in cell death that was identified that linked abnormalities in cell death with human disease was the BCL2 gene. So in 1988, in tissue culture, BCL2 carried by a retroviral vector was put into bone marrow from transgenic mice overexpressing MYC. Now, I mentioned before that these MYC transgenic mice don't develop lymphomas right from birth. And so if you take out the blood cells from these mice before they've developed malignancies, you can then add in extra genes to see if they would complement the MYC gene. And so these are some experiments I've copied out of my lab book. And in these experiments, I've taken bone marrow from normal mice and MYC transgenic mice, and then I've infected them with an empty virus or a virus carrying BCL2. And if the cells had activated MYC alone or activated BCL2 alone or neither gene, then the cells died out in tissue culture. This is a log graph. You can see it's going up orders of magnitude. But if cells had both MYC and BCL2, then eventually the cells would grow out. And this happened again and again, but only with cells that had both MYC and BCL2. And furthermore, if these cells were taken, these cells in tissue culture were taken and injected into mice, then they caused leukemias in mice. So this showed that if you combined BCL2 plus MYC, you could generate immortalized cell lines that caused lymphomas when injected into mice. So this was the first direct experimental evidence that BCL2 was oncogenic, but BCL2 couldn't cause cancers alone. It needed to synergize with another gene such as MYC. But again, MYC couldn't cause cancers alone either. Now, these experiments were done in tissue culture, but a more elegant way of doing these experiments were by using transgenic mice. So we created transgenic mice expressing the BCL2 gene and crossed these mice with mice expressing the MYC gene. You can see that if mice only have the BCL2 gene, so they've got an extra copy and it's an active BCL2 gene, the cells all survive very well, but none of these mice by this age have developed leukemias. I mentioned before that the transgenic MYC mice all develop lymphoma, and half the mice develop it after about 10 weeks. And you can see it's another 10 weeks and half the remaining mice have. But if mice have both a BCL2 transgene and a MYC transgene, then the mice develop tumors much, much more rapidly. So once again, this is showing oncogenic synergy between these two different genes. Now, in 1927, Hermann Muller showed that X-rays could cause mutations in fruit flies. So this again is evidence of an environmental factor causing chromosomal changes, genetic changes that can cause cancer. And in the 1940s, it was shown that carcinogenic agents, alkylating agents, could also directly damage the DNA to cause mutations. Auerbach and Robertson also showed that polycyclic hydrocarbons, so the things in cigarette smoke, in the tar of cigarette smoke, could also bind DNA and mutate it directly, linking mutagenesis to cancer. And also cigarette smoke contains polonium that produces alpha particles that can also cause mutations in the DNA. Now, I see I'm running over time, so I better hurry along. Henry Lynch was looking at inherited predisposition to colon cancers, and Jim Cleaver was looking at other predispositions to cancers with patients with other mutations, and it was shown that in both cases, these mutations were not to growth promoting genes, not to tumor suppressor genes, but these are mutations in DNA repair genes. And as I mentioned before, every cell that divides in two has to make 6 billion DNA bases, has to copy 6 billion DNA bases. And so cells have with them, because the DNA polymerase can make mistakes, the cells within them have mismatch repair and editing mechanisms to repair mistakes when they're made. And so these observations led to a hunt for human genes that resembled bacterial mismatch repair genes, and a number of these were found, including one that was inherited and explained the Lynch syndrome, the inherited predisposition to bowel cancers. Similar efforts underway looking at inherited predisposition to breast cancer led Mary Claire King to hypothesize that there was a breast cancer gene, and when the gene was cloned, it turned out to also be involved in repairing damaged DNA. And over at WeHi, and my colleague Andreas Strasser and Anna Janik showed that one of the several ways that P53 suppresses tumor development is by turning on genes that can repair damaged mutated DNA. So lessons from these experiments is that cancer is initially caused by changes to DNA in a single cell. And a cancer is a clone of cells that generate subclones that bear extra genetic abnormalities. The DNA changes could be due to a virus, a chemical, radiation, or just bad luck, an error in the copying mechanisms. More than one mutation is needed for a cell to become malignant. For a tumor to grow, the rate of cell division must exceed the rate of cell death. Mutations that activate growth-stimulating genes will promote oncogenesis. Mutations that inactivate growth-inhibitory genes will promote oncogenesis. Gene changes that prevent cell suicide will also promote oncogenesis. Loss of function mutations to DNA repair genes will promote oncogenesis by increasing the persistence of mutations. And the human genome, as I've said, has 6 billion DNA base pairs, and over 1 million cells copy their DNA to divide in two in our bodies every second. So about 10 million billion DNA letters are copied every second. So it's quite remarkable that we don't develop tumors all the time, and only about a third of us will develop tumors during our lifespans.